Genomic Sequencing and Human Genome Project. Before looking into what is genomic sequencing, we need to understand what is a genome. A genome is an organism's complete set of DNA. So what is the DNA made up of? So here we can see DNA is made up of a sugar phosphate backbone and nitrogen bases. So this is the structure of the DNA. So this yellow thing here, if we see, it is constant. It is a sugar phosphate molecule that are connected. So what is the varying portion of DNA? The varying portion of the DNA is the nitrogen base. So we can see there are different types of nitrogen bases. That is, here if we see A, adenine, C, cytosine, T, thymine, and G, guanine. So these are the different base pairs, and the combination or the sequence varies, that brings the differences in the chromatin material. So these differences are what brings the difference in the expression of genes. For example, if we see there is a base sequence like guanine, thymine, adenine, cytosine, and the corresponding base pairs. This is a gene. Let's say this is a gene. This produces certain protein that will give some cellular function. So the sequence of these nitrogen base pairs determines the protein that is produced from this gene expression. So this sequence is very important in the DNA of any organism. So if we see, a genome is an organism's complete set of DNA. So we know that a human cell has 23 pairs of chromosomes. So 23 pairs of chromosomes. This is a pair. We have 23 pairs like this. And each chromosome will have chromatin. And each chromatin thread is made up of millions of base pairs. So this sequence, this entire sequence of 23 pairs of chromosomes is the genome. So now that we have understood what is a genome, we will go into what is genome sequencing. So we understood that the arrangement of the nitrogen bases gives that particular protein, or that determines the function of certain gene. So it is very important that we understand the sequence in which these nitrogen base pairs are arranged in a chromosome, are arranged in the DNA molecule. So in genomic sequencing, what we essentially do is map the entire structure of DNA, as in we will particularly look into what is the sequence of nitrogen bases that are in the DNA molecule. Deciphering the exact order of base pairs. So the exact order in which the nitrogen base pairs are arranged in the DNA molecule, we will find out via genome sequencing. This is very important because the arrangement of the nitrogen base pairs determines the protein that is produced from the gene, or the function that particular gene performs. So there are a lot of projects that have been done to sequence the genome in a human chromosome or in human cell. So we will start with Human Genome Project. So this is an international effort that has been made to map the entire genome of human being. So Human Genome Project, it was started in 1990s to map the base pairs, the sequence of base pairs, that is genomic sequencing of the entire human genome, that is all the 23 pairs of chromosomes, that is 46 chromosomes. It has been completed in 2003. So this is the first such project and it is an international project. So recently genomic sequencing is in news because there was another telomere to telomere consortium. This is also an international collaboration. It is said that it has sequenced the first complete human reference genome. So we have already seen that Human Genome Project has done this in 2003 itself. So what is the speciality of this telomere to telomere consortium? So if we see, the Human Genome Project has only mapped 92% of the human genome. That is, if there are 100 base pairs in the entire cell, it has only sequenced 92 base pairs. That is only 92% of the chromatin. This Human Genome Project has left out 8% of the chromatin. Why is that so? So, if we see, this is the chromatin in the cell, human cell, it is said that there is two types of chromatin, that is euchromatin and heterochromatin. So euchromatin and heterochromatin. What is the difference between euchromatin and heterochromatin? Euchromatin, U means true. So euchromatin is major part, and it was believed that this is the most important part of the genetic material. That is, it forms about 92%. This heterochromatin also was called junk DNA, was the 8% that was left out in the Human Genome Project. So Human Genome Project mapped the euchromatin part but has left out this 8%, that is the junk DNA. So recently this telomere to telomere consortium has mapped even this last remaining 8% of the human genome to give the first complete human reference genome. So for exam point of view, we need to know what is euchromatin, what is heterochromatin, and heterochromatin is also called junk DNA. And here telomere to telomere consortium is the first to give a complete human reference genome. So this brings us to the next important point. What is a telomere? This might also be asked as a prelims question. So this is the chromosome. So this ending parts, this edges, the tips are called telomeres. These edges are called telomeres. So these telomeres, telo means end. So telomeres normally consist of repeating sequences of DNA. So this was initially considered that it is heterochromatin or junk DNA, but recently it was discovered that even these telomeres or that junk DNA also determines how the genes are expressed. So even that junk DNA is important in the functioning of a human cell. So that is the reason why this T to T consortium has mapped even the last remaining telomeres or the 8%. So if we see, these two are international efforts to map a human genome. Now we will look into what are the Indian projects that have been done to map human genome. So now we have to understand, why should India do a human genome sequencing if it has already been done internationally? It is because there is a lot of diversity both in terms of race and ethnic diversities between the world or reference world population and Indian population. We all know that India consists of diverse races and ethnicities. So it is important for India to have its own human genome project so that the different diversities can be mapped. and be known. So the first Indian project is the Genome India project. So for prelims examination, this is very important. The Genome India project, it is a collaboration of around 20 institutions. These 20 institutions include IASC and even some IITs. So in Genome India project is an earlier project. It is a collaboration of 20 institutions. What is the aim of this Genome India project? The Genome India project, the aim is to build a reference genome. A reference genome, as in they have evaluated or sequenced genomes of different races and from different places, so that we can have one single reference genome that comprises of all the differences or the diversities in the Indian population. So Genome India project, what are the institutions and what is its aim? This is important for the examination point of view. Next comes Indigen Genome project. So this is the recent one, and this is conducted by CSIR as well as CCMB Hyderabad. This is Center for Molecular Biology in Hyderabad. So these are the institutes involved in Indigen project. And what is the aim of this project? The aim is to undertake whole genome sequencing of thousands of individuals representing different ethnic groups of India. So in this project, basically they have taken up to sequence the genome of different races or different populations in the Indian subcontinent. So we need to know the differences between these two projects, what are the institutions involved, and what are the primary aims of these two projects for examination point of view.